we're going to go ahead and get started. Hi, everyone. So my name is Maritza Garcia. I'm one of the directors of business development for Certified Laboratories. I'm joined by Lisa Elshal. She's our senior director of drug and device consulting from EAS Consulting Group, as well as Rasha Abouid. She's our R&D supervisor for cosmetic OTC and dietary supplements based out of our Carrollton site. And today we'll be discussing understanding feasibility and method validation and verification for OTC and dietary supplements. So we get asked frequently, what are feasibility, method validation, verification, and what are the differences? Feasibility, as mentioned here, is usually done on a dietary supplement when we're wanting to confirm that our method works well with your product. So here we have it listed as a test for a new or unique matrix that has never been tested with us before. It assesses if the analyte can be properly extracted, detected, measured in the product matrix. When submitting to certified laboratories, you can expect our team to perform the testing 3 times to confirm the method and sample are compatible. For method validation, which is usually performed for OTC methods, we're looking to ensure the lab can confirm the test is accurate, consistent, and reliable. So once the method is validated, we can then perform method verification, testing for a specific sample matrix. We're looking to see that the method performs as expected on the specific formulation, sample type, or matrix, ensuring the method is suitable and provides consistent results. So what is a matrix versus a formulation versus a sample? Here we're talking about OTC and dietary supplements. The matrix, as we have listed here, is the physical chemical makeup of the product. So we're looking to see, is it a gummy? Is it a powder? Is it a tablet? Are we talking about a cream? The sample matrix is important because we'll then know how to process the sample or properly extract when performing testing. When we're talking about the formulation, we're talking about the product itself, the full recipe of ingredients, what is in the product. And when we're talking about the sample, we're talking about the specific unit tested in the lab. We'll be using these 3 terms throughout the presentation, something for you all to keep in mind as you're listening and following along. So again, we have 2 examples down here at the bottom of the slide. When we're talking about an OTC, we're talking about an SPF product. The matrix is an emulsion cream. The formulation is an SPF lotion with UV filters and excipients. And the sample is 1 product batch of the SPF lotion. In a dietary supplement, we're talking about a vitamin C gummy. The matrix is a gummy. Gummies can be complex. They can be a sticky matrix. So it's important to extract properly when we're performing testing. Again, another reason for performing feasibility. In this example, the formulation then of vitamin C with pectin, sugars, and flavors. And the sample is going to be gummies with a specific lot number, the product that you're actually submitting over to us for testing. So how do certified laboratories determine whether feasibility, validation, or verification is needed? For new dietary supplements, it's a new analyte or ingredient that we're testing for you. Let's say you're a new customer or an existing customer, but we've never performed testing for your vitamin C gummy product. We're going to want to start with feasibility testing, making sure our method works well with your product, and then report our data over to you, and just confirm again that we don't need to do any additional method development work for your specific product. When we're talking about a method we've never performed, we're going to want to start with a method validation. So this will be, let's say, for a new sunscreen UV filter that's brand new and has just been approved, and we currently do not have a method for it, we will go ahead and perform a method validation for you. That's going to include all the testing that falls under a validation: accuracy, precision, specificity, linearity, range, limit of detection, limit of quantitation, robustness, ruggedness, and force degradation, depending on the method. If we already have a validated method, we're going to perform a method verification, which includes accuracy, precision, specificity, and force degradation. But let's say you already have a method verification for your product, you've already verified it, but you do any kind of reformulation, you do need to perform another method verification to make sure the method is still going to work with the sample matrix and the changes made to that formulation. For existing compendial methods, such as those found on USP, we would perform those for your raw material or product as long as a monograph is available for that specific type of sample matrices. I'm going to hand it off to Lisa L. Shaw. She's going to discuss industry standards and regulatory frameworks for method validations and verifications.
Thank you, Maritza. So why do we have to do all of this? Because it's a regulatory requirement. We're not just trying to make your lives difficult. It is really important that the data that's generated supporting every single product batch release and stability study is as well defined as possibly can be. So the regulations that govern the drug requirements are a couple here. In the 21 CFR Part 211 165E, it says explicitly the accuracy, sensitivity, specificity, and reproducibility of test methods employed by the firm shall be established and documented. Rasha will talk a little bit about how we do that in the technical section of the presentation. 21 CFR Part 211 194A2, suitability of all test methods shall be verified under actual conditions of use. That means, I've got a method, but does it really work for my particular product? So as Maritza mentioned, if we're using compendial methods, they must be verified. Users of compendial analytical procedures are not required to validate the procedures when they're first used in their lab, but documented evidence of suitability should be established under actual conditions of use. And if you're developing your own methods, they must be validated. And one important phrase on this page is the documented evidence. It's really important for all the analytical method development and validation work that you do, that you have the documented evidence to support the testing that you're using in your firms. So the requirements for method validation are defined in the ICH-Q2 and FDA guidance. I mean, the ISO standard, the ICH standard is basically incorporated into the FDA guidance. It's basically the same document. The USP in Chapter 1225 also defines the requirements for a method validation. And then there is an ISO standard 17025 that also defines some of the requirements, but it's really not as detailed as the ICH-Q2 FDA guidance. So when it comes to dietary supplements and OTC products, the label claim verification is really very important. In 21 CFR Part 111.320, it asks what requirements apply to lab methods for testing and examination, and this is the dietary supplement regulation. You must verify that laboratory examination and testing methodologies are appropriate for their intended use, and you must use and identify and use an appropriate scientifically valid method for each specification. So how do you do that? Through the method validation, verification, and feasibility studies that we're already talking about. There's also the Association of Analytical Communities, AOAC, which is an excellent reference. And the key point is you need to be able to support your label claims in the dietary supplement world, especially at expiry, and in the drug world, the label claims that are on the drug facts panel. And so in these examples, you can see in a dietary supplement, the ARIDS 3 is a multivitamin. It lists what's in the product and the amounts. You'll be expected to support that if the agency came to pay you a visit. And again, in the drug world, it's really important that your drug products contain the labeled amount of active ingredient, because that's what gives you the effect that you're looking for. So with that, I'll turn it over to Rasha, and she will take us through the details on how we get all this work done.
Thank you, Lisa. Let's talk about some technical details in here. First, we're starting with what's the difference between a method validation and method verification. Method validation as a purpose is just a demonstration that this method is accurate, reliable, and suitable for its intended use. While in method verification, we're confirming that this existing validated method works for that specific OTC products or formula. In a scope, method validation, we're doing a full evaluation of linearity, accuracy, precision, intermediate precision, and robustness. While for method verification, we targeted all the aspects that's related to the sample, like specificity, accuracy, force degradation, and precision. In the matrix, method validation is being done for one specific or few defined matrix during the validation, while in method verification, it's a product-specific assessment for that specific OTC product or formula. When do we need to use either one of them? Method validation is used whenever we're working with a new method that we do not have any applicable validated method before. While method verification, we use it whenever we're doing reformulation, line extension, or a new matrix formula. The outcome of either one of them, method validation, we're going to have a fully validated method for the defined matrix and analyte. While method verification, we have a document confirmation that this method is suitable for this specific OTC product. Now, how does all of these relate to each other? Validation, feasibility, and verification. The foundation that we're building everything on top of it is a validation. So we started with a fully validation record that's been for this analytical method. On top of it, we built a mini validation called verification. And then on top of that verification, we built the mini verification that is called feasibility, kind of a quick assessment just to see if this method that I have for this active will work for that formula or not. Now, based on the ICH and the FDA guidance, here are the 7 key criteria that are used in analytical method validation. It's kind of a quick confirmation of this analytical method will be performing accurately and consistently every time we're going to use it. Now we're going to be looking at specificity, selectivity, linearity, which include limit of detection and limit of quantitation, accuracy, precision, intermediate precision, and robustness. Now, in method verification, we're going to be targeting all the aspects related to the sample. So method verification is just use this underpinning method validation with confirmation of all the aspects related to that sample. Like we're going to be looking at system stability, specificity, selectivity, linearity for the active calculation, accuracy, precision, intermediate precision, and force degradation in case it's applicable. Example of instrument that we can use in method verification, we can be working with an HPLC, could have been a UV, RI, or MS detector. We can work with a GC, either FID, TCD, or an MS detector. For heavy metal, usually it's tested by ICP-MS or ICP-OES. We can use an UV visible, like the colloidal oatmeal method. We can use an IR, like for dimethic cone methods. And also we can have some method that goes with a wet bench method, just like a titration. Now, compendial methods. Compendial methods are standardized, validated analytical methods. They are published in these official pharmacopoeia, and they are used for testing pharmaceutical raw material and finished products. Mainly, it's used for raw material, but there are a couple of finished products also included in these methods. Some examples of these compendial methods could have been USP monograph, EP monograph, Japanese monograph, or ISO methods. Now, compendial methods are considered to be validated by the time it's already published. That means we do not need to rerun this validation in-house. On the other side, what we're going to do? We're going to do a verification, which is confirmation that this compendial method is suitable for its intended use at this specific laboratory. I'm just confirming that my lab, my reagents, my instrument, and my chemists are able to meet all the system stability requirements listed in this compendial method. I have here some examples of 4 different products. They all have the same active acetaminophen. Now, if I'm having acetaminophen raw material with a purity of 98 to 102, this will be tested using the acetaminophen raw material monograph. While if I have an acetaminophen oral solution, capsule, or tablet, those will be tested by their corresponding USB monograph as long as a product meet the acceptance criteria of the monograph. For example, here I have it listed as 90 to 110% recovery of the label claim. What does that mean? If I have a raw material of acetaminophen that is at 95% purity, we're not going to be able to test it by USB monograph as it doesn't meet the monograph acceptance criteria. On the other side, I will be using my in-house validated method for that sample. Now feasibility is done. Let's look at the report. What are we going to expect to see in these reports? Feasibility as being a quick assessment if the method going to work or not. It's a lot similar to the routine survey with a little bit more details in it. We're going to be looking to see what method number is going to be used, what's the acceptance criteria, what was the final results of these 3 replicates, what is the statistical analysis, relative standard deviation of these results, and any limitation in case it was in this feasibility. While method validation and verification report having a lot more details. They're going to have method description, instrument details, acceptance criteria, summary of the data, statistical analysis, matrix justification, measurement of uncertainty, and conclusion. Let's talk about linearity in a little bit more details in here. Now, linearity is defined as a proportional relation between the amount of analyte I have with the signal that I'm getting from the instrument. I will be using this relation to build a calibration curve as shown here in this screenshot, where I will be able to identify my slope, intercept, and coefficient of determination. Also, based on the 7 key criteria of the ICH guidance, we should be reporting the LOD and LOQ. LOD is defined as the lowest concentration of the analyte that I'm going to be able to see a signal in my method. While LOQ, it's the lowest concentration of the analyte where I'm going to accurately quantify this analyte. Now, some other parameter that would have been included in the method verification report, we'll be having a verification parameter, which is acceptance criteria. Those acceptance criteria will be coming from 2 sources. First, the underpinning validation that we're building this verification on top of it. And the second is a sample itself, the sample submission form. If the sample, for example, have acceptance criteria of plus minus 5% of the label claim, this is what we're going to see here in the acceptance criteria in the method verification report. We're going to be looking at the method details, so you will be able to see all the details or how did we actually perform this method verification. How was the reagent prepared? How did we make the standard stock? The calibration is standard. How did we prepare the sample? What diluent was used during this method? We'll also be seeing the instrument details, so you can see which instrument was used during the method verification, what detector was used, what was all the details related to this instrument in according to instrument parameter. Now, in relation to the samples and results, we're going to be looking at specificity. Specificity can be defined as a quick confirmation that the signal I'm seeing from this sample is only coming from my analyte. How do we evaluate that? We're going to be testing product-specific placebo, which technically should have everything in the sample except my analyte. So the ideal situation that I'm not supposed to see any signal for that placebo. Or if I'm seeing a signal, that signal should be less on my LOD of this analytical method. We'll be also looking at intermediate precision. It's where the sample will be tested 6 times by 2 independent operators. I will be looking at the relative standard deviation of each operator individually and also the RSD across both operators. Force degradation, as everyone have been asking about it, and it's been a hot topic recently, it's just a confirmation that this analytical method is a stability indicating. What does that mean? It means that when this product is going to go in stability and it does change with time, my analytical method will be able to see that change. How do we actually do that in the lab? We force the sample to degrade immediately by applying some harsh condition, and then we're looking at the recovery of these stressed sample in comparison to the control, which is a sample that was not forced into any stressor. We will be also looking at the chromatogram of the stress sample. You see if we're able to see any new peak that were not observed in the control. Now, to mention here that force degradation is not applicable for all analytical method and all actives. For example, in heavy metal, whenever I'm testing zinc or titanium, we do not apply force degradation, as whatever harsh condition you're going to apply to the sample, you're not going to be able to degrade zinc or titanium. And now we will be going back to Maritza.
Thanks so much, Rasha. So why does this matter to you, to your brand, or if you're a contract manufacturer, why should you be doing this? Method validations and verifications can help support CGMP-aligned testing practices, define the label claims with scientifically sound data. Remember everything that you're claiming on that back label. If there's a percentage there, if there's an active ingredient, you need to confirm that you actually have that amount within the product. You're looking to reduce compliance and quality risk, strengthen data reliability and documentation, and protect consumer trust and brand credibility. For feasibility, you're evaluating the matrix complexity. Again, we have gummies listed here, but we're not just gummies, but botanicals, soft gels, tablets, confirming the technical suitability before routine testing, reducing the risk before scaling production, and support scientifically valid testing approaches. I just want to make a quick note here. We just want to let everyone know we've received your questions and we'll have a Q&A at the end of the presentation, so please stay through the end. I am going to now provide everyone with a brief overview of certified laboratories, our locations, and testing capabilities. We have 5 locations strategically placed throughout the US and Canada. We have 2 locations in Southern California, our Tustin site, which specializes in dietary supplements, and Burbank, which also specializes in dietary supplements, cosmetics, and over-the-counter drugs. We also have our Carrollton and Melville sites in Texas and New York with similar capabilities to our Burbank site. For those of you with testing needs in Canada, our Edmonton site can assist with NHP, cosmetic, and over-the-counter drug testing. Our labs are ISO 17025, Health Canada approved, and we've recently become certified through the European Medicines Agency at our Carrollton and Melville sites. And of course, certified laboratories, so the world can trust in what it consumes. That's what we follow. That's what we hope for our industry. That's what we want to make sure we help and support. Additional testing capabilities. We've talked about feasibility, verification, validation testing, but we have a wide range of microbiology, chemistry, and stability testing capabilities as well. For micro, we perform USP 51, PCPC, ISO methods for your preservative challenge testing needs, along with routine USP 60 B-Cepacia complex, USP 61, 62, which includes TPC, yeast, mold, and pathogens. If any growth is found, we can assist with the identification testing as well. For chemistry, we can perform active OTC testing. So we're talking about sunscreen actives, salicylic acid as an example, preservatives such as formaldehyde, phenoxyethanol, sodium benzoate. And for contaminants testing, that includes heavy metals, pesticides, residual solvents. And of course, we test water and fat-soluble vitamins as well. For stability, we're performing testing under ambient and elevated temperature conditions with humidity, and we can include packaging compatibility and photostability as well. Our specialized test includes CBD and THC, sterility, colloidal oatmeal, and gummy and soap matrices, as well as Amazon testing. We are an approved tick for ophthalmic, skin brightening, and dietary supplement products. So you can reach out to us for more information on those. So next steps, please do reach out to our certified labs team for your testing needs. Any questions on pricing, onboarding requirements. We've included the contact information of our sales team. So here we have Todd Shea, our president, Stephen Taormina, our vice president of sales and marketing. Again, my name is Maritza Garcia. I oversee the Southwest. We have Michael Sylvia in the Northeast, David Cook in the Central West, Carla Wright in Central, Alan LaCroix in Southeast, and Ashley Trevisani in the Northwest and Canada. We're now going to proceed with Q&A and try to answer as many questions as possible. For any remaining questions, our team will reach out to you after the webinar. I also want to mention now that there will be a brief survey at the end. We ask that you please take a moment to answer the questions. Lisa, we'll start with you.
Great. Okay, thank you again, Maritza. I'll start with some of the questions that we received ahead of the webinar. So one is the differences between the US, Canadian, NHP, EU, and UK requirements. Again, I think the best thing to do is to consult the regulatory agency to make sure you've got the right standard. But the ICH Q2 document really is the most comprehensive, and should, if you're following that and you're meeting those requirements, you should be your method validation work should be acceptable to all those different regions. But again, it's always a good idea to verify that before you just go on and say, okay, I'm going to be fine. Just always do that check. The next question, criteria for scientifically validated. I think Rasha went through that in pretty good detail during the course of the presentation. She talked about all the elements of a validation, and depending if it's a full validation, a verification, or a feasibility study, which elements apply. And again, as far as documentation goes, I would retain the reports, the notebook references, all the data that you've generated in the course of validating the method, any change controls, the methods themselves, any what's the word I want to use? Any types of deviations, any justifications. You really just want a good comprehensive package of information to support the products that you are putting out on the market. Again, what are the responsibilities of a contract manufacturer? So as a contract manufacturer, you're responsible for the quality of the products that you're producing. So the FDA will require you to have validated methods for the products that you're testing. And they're going to want to see the evidence of that validation. And it doesn't matter if the client that you're making those products for tells you that they don't need that. The FDA will hold you as a contract manufacturer accountable for the quality of your products. So it's in your best interest to ensure that all your testing is done under validated methods. And I'm using the word validated, but it could be feasibility, it could be verification. I'm just kind of using that as the extreme highest level standard. Another question, if the founders are physicians, does that change anything regarding testing and regulations? And the answer is no. It doesn't matter who you are, what your role is. At the end of the day, all products are required to follow the same quality standards as defined in either the drug GMPs, 210 and 211, or the dietary supplement GMPs. So the consumer who's purchasing your product, whether it be in a brick and mortar store, on Amazon, or wherever, expects the same level of quality regardless of who's responsible at the company for manufacturing that particular thing. The next one, how to verify and test my products to make sure it's safe to sell to the product, to the publics, excuse me. So this is where you could work in conjunction with the certified team, with a consultant group like ours. It all depends on what your product is and defining what those critical quality attributes are to ensure that you develop robust product specifications and that the testing that you do supports those specifications. So there's not really a black and white answer. It just kind of depends on your product type. And then the last one, for OTC products such as alcohol hand sanitizers or medicated wipes, how should QA teams determine whether the analytical method used by a third-party lab is sufficient to substantiate label claims for the active ingredient concentration and efficacy? So in this case, as if you're using a third-party laboratory, you saw the examples that Russia provided about the reports that you're going to receive based on the work that they do. You want to make sure that you're reviewing those reports and that all the elements that you feel are important to your product are the elements of the validation are addressed in all the work that's being done by a third-party lab. So with that, I think now we can move to the questions that have been submitted as part of this session. Oh, no, sorry, Rasha, I know you wanted to address, you had a few you wanted to answer first. Sorry about that. Go ahead.
Okay, thank you, Lisa. So now I had a couple of questions previously submitted. Let's go over them. First question is, and that's a pretty often asked questions for us. Let's say I'm using, I have a product, sunscreen product with avobenzone homosalate, and I have 20 shades of that product, from dark to medium to very light tan. Now, the base formula of these products are exactly the same. It's just a minor difference in the shades. Or, for example, if I'm having a toothpaste, could have been a minor difference in the flavor of that toothpaste. Do I need to run 20 method verification? That doesn't seem that much practical. So what we do in this case, that we're gonna pick the darkest shade, the one with the most flavor, and we're gonna assume that's my worst case scenario. I'm gonna apply my full method verification into that product, while all these other variation of the color, of the flavor, of the shade will be added also in the verification as a specificity inclusion. But the full method verification gonna be done for the worst case scenario, the one with the most product or the one with the darkest shade. And we're assuming if it's gonna work with this one, it's gonna by default work on all of these other included products. The other question I have here, what if the method validation or verification fails? And that does happen. For a specific product, let's say I'm realizing that I'm having interference. I have another peak that's coeluting with my active, which affecting how accurately I can actually quantify it. So what are we going to do? Certified lab have a very qualified innovation team. We're going to move this method into some R&D department. We're going to do a lot of R&D work on it. We're going to need to modify anything in the method so that we're able to accurately quantify and separate this active from any interference. One other question we had is about method transfer, and I've seen it also in this live sessions too. Now, let's say you have been doing stability for a while somewhere else, you have a validated method, and you would like to keep using this method. So what we're going to do is we're going to be running an official method transfer. We do not revalidate that method. We will just look and review that old validation report that you have. And then based on that, we're going to run a method transfer so that we can accurately adopt this method that you have been previously working with. And that is good for me. Maritza?
Thanks, Rasha. I'm going through some of the questions that we've received. Some of these are in regards to suitability. I assume that this is for micro USP 6162. We do that, as mentioned in the presentation at our sites. It is a USP requirement, so something for those of you asking about that to keep in mind. We'll go ahead and have someone from our team reach out to you with additional information. Any other questions that we're going through the chat live right now, so looking to see if our team can answer any additional questions. Rasha, maybe you can help answer this one. Is method transfer the same as verification? I think you just talked about that. And shaking your head no.
So there is similarity in method transfer and method verification. Both of them are built on top of the originally validated method. In method verification, I'm just confirming in the same exact lab, in the same exact condition, that I'm able to accurately test this product. Method transfer is usually done between two different locations. So let's say I have a validated method in a previous lab, and I want to adopt this method in one of our current lab. So we transfer this method. What we're going to do is we need to make sure that I'm accurately able to meet all the system stability requirement. And we do have some kind of equivalency between the original lab, where the original validation was done, and then the receiving lab, the lab will be adopting this method.
Thanks, Rasha. I believe you covered this during your presentation, but on your portion, but why do we do force degradation?
So force degradation, we need a proof that whenever this product, when it's going on stability, we're going to be actually seeing the change and the degradation of this analyte with time. So without that, how are you going to prove that? What if your product is changing with time and your method doesn't seem to be sensitive enough to see that change? So this is where force degradation is coming from. It's a proof that this analytical method is a stability indicating. But force degradation does not replace the need of doing a stability study. It's just an evidence to support that stability study.
Thank you, Rasha. And I'm going to ask this next one because it sounds like this is one that you can also answer. Lost it for a second. So if a compendial method verified already and with a slight change of raw material, sample prep is slightly modified, like add EDTA, for example, does this need a full verification again or just feasibility?
Now, USP, for example, as a compendial method talking about USP, they do have a Chapter 621 chromatography that listed what are the allowed modification to the monograph where you do not need to consider it a totally different method. If these changes are done within the allowed acceptance criteria of the monograph, for example, the monograph allow you to change the mobile phase pH plus minus 0.2. So if your change is within plus minus 0.2, then we're okay. We're still within the USP acceptance criteria. But if these changes are done outside of the acceptance criteria of the USP monograph, then we're not going to be able to call it USP monograph anymore. We have to give it an in-house number, and we have to validate this modification. And I guess Lisa can confirm from a regulatory point of view.
Yes, that's correct. Absolutely.
Okay, let's see.
I mean, I can answer the one about the OTC stage product. Do you want me to take that one, Maritza?
Yes, please, Lisa.
Yeah. So the question is, what type of OTC stage product should be sent for method verification suitability? The question is, can it be a lab R&D sample or bulk sample or finished product sample, specifically for the micro method? And what's the FDA requirement in that aspect? Well, the FDA doesn't really tell you exactly where in the stage of, your product development cycle that you should do your validation testing or what kind of sample. But in general, and my background is OTC product development. In general, you don't want to start method validation until you finalize the formulation, because you want to make sure that what your method validation is supporting is the final product as close as possible in the package, in the final package, or something really close. So I would say once you've got approval from whoever it is that decides that this is the final product, and marketing, who's famous for changing their mind at the last minute, doesn't say, oh no, I wanted purple, it's green, but now I want it purple, and it used to be, maybe lavender fragrance, and now we want green apple or something crazy like that. So make sure you've got the final formulation, and you can use any of those batches. I mean, it depends. Some companies have policies where they only want to do the validation work and formal stability on a GMP batch. Some, as long as the formulation is representative of the final product, you can use a lab scale batch. The point is, you don't want your method validation results to be skewed by the product not being manufactured correctly. Most of the time, the method is really dependent on the components of the formulation and not necessarily how they were put together. So, for example, when we do placebo interference specificity studies that Rasha was talking about, those are typically done on a placebo that's manufactured on the lab scale. We're not going to go into a manufacturing GMP site to make a batch that doesn't have the actives in it. So that would be an example. But again, you just need to be, as I said, back on the regulation page, be really clear about what you're validating. Make sure that you've got that formulation documented. And if there are any changes that are made after the fact, you need to do an impact assessment to determine if you have to go back and revalidate any of the elements that you might have, that might have been impacted by the change in the formula. So does that capture anything that you've seen differently, Rasha, with your group?
No, that does sounds good. Thank you.
Thank you.
Okay, and we have one more, Rasha, that I think you could answer. Could force degradation tell you what degradants you can expect to see?
Okay. So force degradation, we're going to be able to see any new peak that's been observed after the sample being stressed out. We're not going to be able to identify what is exactly that peak. Unless I know exactly what to expect, something I can put in to confirm the retention time, but we usually just record the number of peak observed and the retention time of these peak observed, but we do not identify those degradants unless I know for sure that I need to look for that specific product as a degradant, then I can add it in my sequence and confirm retention time. But in general, we're not identifying what is that degradant, we're just reporting the retention time.
Yeah, and I can add that example that Rasha gave for the acetaminophen. There's a known degradant for aminophenol that I'm sure would be evaluated to see if there's any 4-aminophenol that's formed as part of that forced degradation study. Because the idea, as Rasha mentioned, is you want to be able to analyze the actives in the presence of their degradants.
Mm-hmm.
Thanks, Rasha. Thank you, Lisa. Okay, so we are going to conclude here. We have received additional questions. You will receive answers to these after the webinar. I want to let you know that there is a brief survey at the end. We ask that you please take a moment to answer the questions. It'll help us improve on the content and what we present to you all when we do these webinars. But I really want to thank you all for joining us. The recording will be made available to you afterwards. And again, if you have any additional questions, do let us know, but we will be reaching out to you to answer any questions that we were unable to answer during the webinar. Thank you so much and have a great rest of your day.